And our next speaker is Grace Capshaw. Grace is a postdoc at Johns Hopkins University. She works with bats. And a very fun fact about bats is that they have an unusually long lifespan for being such small mammals, which is very exciting. And I believe Grace will bring that up in her presentation as well.
Yes. Thank you so much, and thank you for your attention. I know it's kind of late in the day, but I am a postdoc in the labs of Drs. Amanda Lauer and Cindy Moss, and so today I'm going to tell you about sort of a collaborative project bridging the two labs about how brain-to-ear feedback might support resilience in changing acoustic environments. So many here are probably familiar with this kind of scene. This is a typical urban environment, really heavily modified by humans. So there's lots of sights and smells and sounds that are associated with dense human populations. And sometimes, depending on the level and type of human activity, these sounds that are generated can be incredibly loud. So for example, sounds that are generated by transportation, so like highways, railways, planes taking off and landing, these can range from 80 to 140 decibels. Things like sirens are meant to be super loud, and those can range from 100 to 130 decibels. And industrial activity, so things like construction, mining, excavation, those can generate sounds up to 150 decibels. And so I realize that not everyone here is an acoustician, so just to kind of give you an orientation to what the decibel scale means perceptually, very faint sounds like wind and rustling leaves and moving water tend to be between 10 and 20 decibels. Human conversation, just kind of two people talking to each other, is about 60 decibels. We start to think of sounds as being loud at about 80 decibels, and 120 decibels is actually the threshold for pain, at least in humans. We don't really know about that in other animals. And so when I present these sounds to you, these particular numbers, it's really important to understand that sounds in nature very rarely exceed 80 decibels. So the vertebrate auditory system hasn't really evolved to experience these levels. It's also important to know that continuous exposure to sounds over 85 decibels, so whether this is chronic sound or like constant acute repeating sound, this can be really damaging to auditory systems. And so we know that most vertebrates rely on acoustic signaling for things like territory defense and mate detection and selection. And for some, acoustic signaling is essential for foraging, so it's sort of the difference between surviving day to day. But the physiological aspects of noise on their auditory systems, and specifically in species that aren't traditional laboratory models, is really less well understood. One thing that we have characterized really well is that nearly all mammals, including humans and some non-mammals, will develop hearing deficits over time and with age. So this figure is just showing the prevalence of hearing deficits as in different age groups in human populations, and you can see a steady increase that becomes sort of precipitous with increasing age. But it's important to know that the trajectory of hearing loss over a lifetime is not just due to natural senescent processes. Some will lose their hearing earlier due to genetics. Some become exposed to ototoxic substances, things that kill hair cells. And importantly, the cumulative effect of a lifetime of sound exposure can be really damaging over time. So for example, overexposure to sound is known to cause mechanical damage to the sensory hair cells in the cochlea, so it can lead to damage to the stereociliary bundles on top. It can also generate excitotoxicity at the synapse, and so the ribbon synapse and the afferent fiber that it communicates with can degenerate, and this will lead to apoptosis of the hair cell, and you start to see cellular loss within the inner ear. So this is just general exposure to sound over a long period of time. But it's important to know that not all species are equally affected by exposure to noise. So some species, like mice, are really susceptible. So here are some results from a recent study on the noise effects on mouse hearing. And just to orient you, this is called an audiogram. On the x-axis are the frequencies of sounds that are presented to a mouse. On the y-axis are the detection thresholds that are physiologically determined. So this is the quietest sound that evokes a response in the mouse's auditory brainstem. And so each curve is an audiogram. So in black is the hearing for a mouse pre-noise exposure, and in red is the hearing for a mouse post-noise exposure. And so you can see almost a 40 decibel threshold elevation after acute exposure to loud sound. And so these mice are essentially deafened. On the other end of the spectrum are species that are more resistant to noise exposure. And so in this same study, bats were exposed to the same noise exposure paradigm, and they found virtually no effect on their hearing threshold. So you can see that, pre and post hearing, it made no difference to the bats. And so there's some evidence that bats might be more resistant to the damaging effects of noise than something like a mouse. And so the main question that's driving my research is, what innate mechanisms confer resistance to the effects of noise in some but not all species? So just quickly to back up and introduce the auditory system for anyone who's not obsessed with auditory processing. Everyone here probably knows the canonical ascending pathway from their Intro to Neuroscience course. Sound energy is received by the cochlea, and it's transmitted as an electrical signal up through the brainstem, into the midbrain, and finally to the cortex. So this is ascending afferent information. What you're probably less well aware of is that there is also a parallel descending efferent auditory system, and this allows higher centers in the brain to modulate activity in lower centers. And particularly of interest to my research is what is called the olivocochlear system, through which efferent projections from the olivary complex in the brainstem project down to the cochlea. And it's actually through the olivocochlear system that the brain can control the cochlear response to incoming sound. So there's two main branches. There is the lateral olivocochlear system. So this sends projections to the afferent fibers that are synapsed to the inner hair cells. And so these can actually modulate afferent activity coming from the cochlea. There's also the medial olivocochlear system, and this projects onto the outer hair cells, so you can see it kind of in red. And this modulates sort of the cochlear gain. So outer hair cells are really important for either amplifying or dampening the response of the cochlea to sound. And so just to give you an idea of what exactly the olivocochlear system is doing, here we can see a cartoon of a sound-evoked rate level function for an auditory nerve fiber. And in this study, they presented a tone at a range of decibel levels on the x-axis, and they recorded the response of the auditory nerve fiber in spikes per second. And so you can see a really nice representation of the dynamic range of this nerve fiber, where firing rate can encode the changing intensity of that tone. Now, when that tone burst is presented in noise, so it's embedded in background noise, the firing rate of this auditory nerve fiber no longer changes with changing tone level. So in this case, you can actually see that the presence of background noise will compress the rate response function of this auditory nerve fiber. But in this experiment, they paired the tone and noise with electrical stimulation of the olivocochlear bundle, and you can see that the rate response is actually restored. So olivocochlear stimulation reduced firing rate to the low-level tone stimuli in noise, and it increased the saturation rate at higher levels, and you can see a restoration of the dynamic range of this auditory nerve fiber to the tone embedded in noise. And so the olivocochlear system is hypothesized to have some super important auditory functions. It's thought to regulate auditory nerve activity during postnatal development to essentially tune your cochlea to sound. It's thought to exert top-down control onto those outer hair cells to turn down cochlear gain in the presence of really intense sound, so this might protect against acoustic trauma. It enhances frequency discrimination and the ability to unmask signals and noise, so this is important for the cocktail party problem that people might experience. And it's also hypothesized to mediate selective attention, in which incoming sound can be filtered at the cochlea to enhance your sensitivity to relevant signals while suppressing the response to irrelevant ones, depending on what you're paying attention to. And all of these functions are essential for maintaining a healthy auditory system that's able to adapt to changing soundscapes, which is why we're most interested in exploring efferent auditory specializations. So why do we study bats? Well, bats are auditory specialists. So echolocating species of bats will emit an ultrasonic signal, and they'll listen for those returning echoes. And so bats are highly sensitive to quiet sounds. But at the same time, bats will often live and forage in groups, which introduces lots of competing sounds, and so their auditory system needs to be able to detect and distinguish sound, signals embedded in noise. And they're also constantly exposed to sound, including self-generated sound. So some species, their echolocation calls can be as loud as 140 decibels. And so there's sort of this idea that there must be some protective mechanism to not deafen yourself or your neighbor. And finally, bats live to an incredibly long lifespan for their size. So some species can live up to 40 years in the wild. And because echolocation is essential for survival, their ability to live to old age presumably should require retention of hearing across that lifespan. And so recent work has shown that noise exposure doesn't induce hearing loss in bats. So I've already teased these results. In this study, both mice and bats were exposed to intense broadband sound for 2 hours, which deafened those mice. And those physiological deficits were correlated with structural changes. So you see a lot of damage to the cochlea, lots of hair cell loss. In contrast, bats showed no change to their hearing thresholds, and there was also no noise-induced loss of those cochlear hair cells. Hair cells were perfectly fine after this noise exposure. And in this study, they actually replicated this in, I think, like 8 different species of echolocating bats, and none of them lost their hair cells when exposed to this noise. And so this indicates that the bat cochlea might have some general mechanism that protects it from the damaging effects of noise. Similarly, it's really well established that mice, like most mammals, including humans, show a gradual decline in their hearing over time. So here you can see a waveform representing the sound-evoked auditory brainstem response, or ABR, in young and old mice. So the black wave shows the evoked response in young mice. So you see those really large, robust waves indicating a good hearing, whereas the older mice in red show very small, diminished, slightly delayed waves. And this correlates with reduced hearing abilities over time. So these are, again, audiograms for male and female mice taken at different time points along their lifespan. And the main take-home is that as they age, they gradually lose their hearing. And of course, these hearing deficits are tightly linked to structural damage. So these plots show that aging mice can lose up to 50% of their hair cells and between 30 and 50% of those presynaptic ribbons that are essential for transducing sound into a neural signal. In contrast, we wanted to look at age-related hearing loss in bats, and spoiler alert, they don't seem to have it, at least this species doesn't. We measured ABRs in the echolocating big brown bat and found no age-related deficits either in the amplitude or latency of those sound-evoked waves. We found no evidence for elevated threshold with age, so when we compared young bats in yellow to aging bats in blue, we saw no sign of hearing loss across the entire range of frequencies that they're sensitive to. And then when we compared cochlear structures in young and aging bats, we found no evidence for loss of sensory hair cells, which are shown here in blue, or presynaptic ribbons shown in green. And so, whereas aging mice might lose up to half of their sensory cells and presynaptic ribbons, there's no change across young and aging bats. So bats seem to retain their cochlear structural integrity even into old age. And so the next question is, what sort of mechanism might be contributing to resistance to noise and aging in the bat auditory system? And we think an important player is the olivocochlear system, given its hypothesized function of acoustic signal unmasking in the presence of noise and also protection against cochlear damage, both of which are probably really important for mediating the behaviors of echolocating bats. And we actually have some preliminary evidence that the olivocochlear system might be hypertrophic in bats compared to mice. So here we show chat-labeled olivocochlear cell bodies in mouse and bat brainstem, and you can see many more cells in the bat. These cell bodies, of course, project to the cochlea, so we wanted to look at that. And when we labeled the efferent terminals in the cochlea, you can see some key species differences, both in the lateral and the medial olivocochlear system, which we then quantified. So we compared innervation patterns in bats and mice, and looking first at the lateral olivocochlear system, you can see that terminal density in mice is about 5 to 15 microns per inner hair cell, whereas in bats, it's about 5 times that amount. For the medial olivocochlear system, in mice, innervation density is about 1 to 6 square microns per outer hair cell, whereas in bats, it's between 4 and 10 square microns. And so whether these differences reflect variation in efferent control of cochlear activity across these two species, and whether this is what is contributing to resistance to noise and aging in bats, is still an open question. But we're hoping to sort of follow these up by answering 3 major questions. First, we're looking to see to what degree do bats maintain their auditory structural and functional integrity following exposure to noise. We're hoping to implement targeted approaches to modulate efferent activity and see how these different feedback loops can contribute to protection to noise in bats. And we're also starting to leverage molecular methods to understand what sort of dynamic changes are occurring in the cochlea and the olivocochlear system following exposure to noise. And so we think bats are a really powerful model to explore mechanisms that might confer resistance to noise, and we believe the auditory efferent system might be playing also an important role in whether a species is able to adapt to anthropogenic noise and depleted soundscapes, which could be the key to survival in human-modified environments. And so I wanted to add this slide to sort of address the open questions that are, or the big gaps that exist. I just want to emphasize that although bats perform a really specialized behavior in echolocation, their auditory system is fundamentally the same as other mammals. And so the challenge that we often have is how do we distinguish between specialization and generalization? And I think the only way we can do this is to incorporate more diverse species through comparative study. So this is the only way we might be able to differentiate between what is a specialization, only bats have this, and what might be translated beyond your model organism to predict how other species might respond to changing environmental pressures. I might be preaching to the choir here because everyone has been presenting really beautiful, interesting, diverse species, but most often when I'm presenting auditory research, the mouse is king, and often you have to sort of justify the bat or really anything that isn't a mouse or a non-human primate. And so I just think it's really important to incorporate diverse species so that we can understand what actually is going on and understand how these might change. And so with that, I just want to thank my 2 labs, in particular Clarice Diebold, who was fundamental for the aging research, and also thank you for your attention.
Thank you. Yeah, first question already right there.
Great talk.
Oh, thank you.
Hi, I'm Diana.
Hi.
Thanks for the great talk. Are there any species of bats where echolocation is less important that you might be able to use as a comparative, but controlling
Yes.
for bats?
Absolutely. So there's the bat I'm using is an echolocating insectivore, so it is heavily reliant on echolocation to eat. But there are a ton of frugivorous bat species, and for example, there was also a recent study looking at age-related hearing loss in the Egyptian fruit bat, and they found that they lost their hearing as they aged. And what's really interesting about that species is that it actually prioritizes visual cues over acoustic cues when it's given sort of the choice between the two. And so you can imagine that species that have access to more sensory modalities maybe can kind of adapt to the loss or degradation of one particular modality, and some species might really it's the difference between eating or not for this particular species that I work with. So there's a whole lot of diversity even just within bats. Certainly there's bats that lose their hearing, and certainly there's bats that don't even echolocate.
Thanks.
That was a great talk. I'm Philip Washburn from the University of Oregon. I seem to remember something about bats being able to uncouple, do mechanical uncoupling while they're emitting their loud sound. And then they kind of switch on their hearing to hear the echo coming back. I just wonder whether that might just explain a lot of it, and that essentially for a lot of the time they're kind of deaf.
Yes, yeah, so I think what you're thinking about is the middle ear muscle reflex, and so this is where when exposed, so even humans have this is sort of a mammally thing. There's a muscle that connects to your stapes, which is sort of the input of acoustic energy into your cochlea through the oval window, and when that muscle contracts, it sort of holds the stapes in place, and that sort of is a reflex that occurs in the presence of loud sound. So if if a balloon pops next to you, you might not realize it, but your muscle has probably contracted just very briefly. And bats do have a phasic contraction of that middle ear muscle as they echolocate. So it's actually really interesting. There was a study that measured the activity in that muscle as the bat was vocalizing, and it does tonically contract every single time that bat emits a call. But this doesn't necessarily work when there are other bats that are emitting in close proximity. So this is probably helping against self-deafening, but it doesn't necessarily protect you against the noise that's generated around you. So I don't have any images of how bats tend to live, but they live in most species will live in sort of a colony. And so there's just tons of bats all clustered together, sort of snuggling and just constantly sort of screaming. And so it might protect against your own self-vocalization, but not necessarily fully protecting against others. But certainly that's contributing. Yeah, we would expect it to.
Okay, last question.
Geez, sorry. Sean O'Donnell from Drexel University. Hi. So I'm going to not say birds. Is there any hope of looking at these kinds of processes in cetaceans? Because, like, to me, that's the mammal system that has some, it's long-lived, relies on echolocation, lots of loud noise.
Yeah, certainly.
Or is that like just too hard to do? Or
I mean, that would be fantastic. It's really, I think, one of the greatest challenges working with, really diverse species is getting a hold of them. And so if you don't have access, because, it's very rare that anyone's euthanizing a cetacean or, when one beaches that you've come upon it before it's been sitting there for a long period of time. And so I think one of the challenges that we come upon when we try to incorporate, really big, hard to access species is postmortem time, which can make then a lot of the anatomy sort of nothing. But we're working on it, and it's definitely a point of interest because certainly I'm working with an aerial echolocator, but there's a lot of anthropogenic sound that's being introduced in the oceans, and this is a huge problem. And so yeah, that would be fantastic if we could look at the efferent system in that species and in cetaceans. That would be amazing if you've got any.
Awesome. Thank you, Grace.
Thank you.
I know there was more questions, but